only convert 10 molecules of substrate to the product, or say 100 molecules of a substrate to the product. An enzyme can convert infinite number of substrate molecules to the product molecules. If this thing is happening, often you will observe that a single substrate molecule is utilized for the purpose of synthesis of different kinds of product molecules by the cell. One amino acid may be required for synthesis of a particular kind of protein. The same amino acid will be required for further conversion into another form of amino acid. Say, for example, if you take the case of say glycerol. Glycerol is utilized by certain microorganisms to form L-lysine. After L-lysine, the same L-lysine molecule will be converted to threonine and methionine. That is the universal pathway occurring in a living cell. एक ही substrate molecule से multiple product molecules तैयार किए जा सकते हैं. The organisms follow a particular pathway for that purpose. Okay? Or in some cases, like if you talk about those organisms which synthesize acid from a glucose molecule. Now, in that case, what is going to happen? The organism will utilize the glucose molecule, it will convert it to some kind of acid, and meanwhile, it will synthesize certain amount of ATPs from that metabolic process. But eventually, what happens as the conversion continues slowly and slowly and slowly, there will be built up of acid surrounding the cell's environment. So consider that organism is growing in one flask. In that flask, you have provided glucose, and if it is converting that glucose to say some kind of acid, so that acid slowly and slowly builds up in that flask, and as a result, what happens? The acid is accumulating. The pH of the flask will start decreasing. आहिस्ता आहिस्ता क्या होता है कि फ्लास्क के कंटेंट्स का पीएच जो है वो एसिडिक हो जाता है. एंड यू नो वेरी वेल दैट हाईली एसिडिक पीएच is detrimental for the growth of microorganisms. So in such cases, what is going to happen? That too much accumulation of acid is going to harm the organism which is forming the acid itself. So in such cases, there should be some regulatory mechanism. Somehow the enzyme should be capable of controlling the extent of reaction to which it is carried out. Means with respect to the number of substrate molecules being converted to the product or like that. Okay, so in that scenario, the enzymes do have a mechanism which is referred to as the enzyme inhibition. Now, what basically this inhibition is? Inhibition refers to a process of stopping something. As you could see, we have placed a stop symbol in this slide itself. Now, this indicates that the organisms have a mechanism to stop a particular process at a particular point of time. Now, this process stoppage will be dependent upon various different factors like the amount of product which is being formed, the amount of substrate present in the media or in the environment where that organism is growing like that. Okay, so inhibition will be a process which stops an enzyme catalyzed reaction. Okay, first we will see some basic concepts regarding enzyme inhibition. The first thing which comes in our observation is the substance, the inhibitor molecule. who can inhibit the enzyme catalyzed reaction. So an inhibitor will be any molecule, any substance that can diminish the velocity of enzyme catalyzed reaction. कोई भी ऐसी चीज, कोई भी ऐसा केमिकल, कोई भी ऐसा सब्सटेंस जो एक एंजाइम कैटलाइज्ड रिएक्शन की वेलोसिटी को तोड़ सकता है, ब्रेक कर सकता है, एंजाइम कैटलाइज्ड रिएक्शन को रोक सकता है, इट विल बी रेफर्ड टू एज अ इनहिबिटर molecule. Now, the naturally occurring inhibitors usually often are the product molecule itself. For example, say हमने एक केस देखा आपके एसिड फॉर्मिंग बैक्टीरिया का कि उसके अंदर उसका एसिड जो है द प्रोडक्ट मॉलिक्यूल इटसेल्फ एक्ट्स एज अ इनहिबिटर मॉलिक्यूल. कब करेगा? एक पर्टिकुलर पीएच जब अटेन होगा रिएक्शन के दौरान में, सो वो थ्रेशोल्ड वैल्यू अचीव होते ही वो प्रोडक्ट मॉलिक्यूल जो उसी ऑर्गेनिज्म ने तैयार किया है, वो प्रोडक्ट मॉलिक्यूल जा करके उस पाथवे की कुछ रिएक्शंस को ब्लॉक कर देगा. एज अ रिजल्ट देयर विल बी नो फर्दर अकोमोडेशन ऑफ दैट पर्टिकुलर एसिड ड्यूरिंग द रिएक्शन. नाउ दिस इज द नेचुरल प्रोसेस ऑफ control. Now, this is usually referred to as feedback inhibition. Okay, later on we'll see in detail what feedback inhibition is. But the same knowledge is utilized for various different purposes also. अभी हमने जो कहा regulation of reaction, it is a natural process, intrinsic process of an organism. But the same mechanism is also carried out or utilized for our benefits. For example, say we utilize various different drugs, antibiotics, antimetabolites, and poisons. Various different drugs are there, antibiotics or antimetabolites are there, which are used to treat certain human ailments. So what basically we are doing, say in case of antibiotics, if you refer to the antibiotics, अगर आप antibiotics को देखेंगे, so in that case of antibiotics, certain antibiotics, they are acting as the inhibitor of some of the enzymes. For example, penicillin, if you take the case, penicillin acts as an inhibitor of certain enzymes which bring about synthesis of cell wall of gram-positive bacteria. Therefore, penicillin is active against gram-positive bacteria. It is not active against gram-negative bacteria. The reason for this is that the penicillin molecule is acting as an inhibitor of gram-positive bacterial enzymes. It is not acting as an inhibitor of gram-negative bacterial enzyme or viral enzyme. So as a result, the penicillin has got that limitation that it will be active against the gram-positive bacteria. Okay, and how is it active? It is acting as an inhibitor of a particular enzyme which is responsible for cell wall synthesis of the bacteria. As a result, gram-positive bacteria are unable to synthesize their cell walls, complete their cell walls, and eventually they die due to lack of complete cell wall. Cell wall का function क्या है? To protect the microorganism, to protect the cell. If cell wall is incomplete due to some reasons, the cell is prone to the environmental factors. Immune system उसे आसानी से kill कर सकता है. Similarly, we utilize certain anti-metabolites. Say कुछ जो diseases है ना, नॉन माइक्रोबायोलॉजिकल जो इन्फेक्शंस होती है, से इंडाइजेशन है या उस तरह की और बहुत सारी जो एलिमेंट्स है, in that ailments, or the best example is the case of your painkillers, whatever the painkillers you use. Paracetamol है आपके, डाइक्लोफेनाक है, निमेसुलाइड है. All these painkillers, they are actually the enzyme inhibitors. What they do, that they go and block the inflammatory enzymes, those enzymes which are responsible for transmission of pain feeling to your brain. So as a result, whenever you take a painkiller, you feel better, that now our pain is diminished, it has gone. आपको लगता है कि पेन किलर खाने के बाद आपका दर्द खत्म हो गया है. You should keep in mind that pain has not relieved, तकलीफ खत्म नहीं होती है. What has been done that these medicines, they have only blocked the enzymes which are sending the pain signals to your brain. Means अब आपके ब्रेन तक वो इंफॉर्मेशन नहीं पहुंच पाती कि कहाँ पर तकलीफ है और किस तरह की तकलीफ है. एज अ रिजल्ट व्हाट हैपेंस, आपको तकलीफ महसूस नहीं होती. तकलीफ होती है जिस रीजन की वजह से आपको पेन हो रहा था, से यू हैड सम क्रैम्प्स इन योर मसल्स, सो वो क्रैम्प्स अपनी जगह बरकरार रहेंगे, बट the pain signals will not be sent to your brain. As a result, we feel that the pain has diminished. Pain has not diminished, pain is still there at its place. The cramps are there, but we have stopped feeling that pain due to these drugs, these medicines. Similarly, there are certain poisons, like in case of diarrhea conditions, when persons suffer from loose motion. So there are certain chemical molecules, heavy metals, or there are certain uh, microbiological metabolites, pathogenic organisms के कुछ metabolites होते हैं, which may act as the inhibitor of certain regulatory enzymes in our intestine or digestive tract. As a result, the whole process gets imbalanced and the person may suffer from conditions like diarrhea and all those things, or vomiting, similar things. Okay, so basically what is happening, that a chemical entity, a chemical molecule is going and attaching to the enzyme molecule. As a result, the enzyme molecule becomes inactive. एंजाइम मॉलिक्यूल की एक्टिविटी पूरी तरीके से खत्म हो जाती है ड्यू टू अटैचमेंट ऑफ दीज सब्सटेंसेस, and such substances will be called as the enzyme inhibitors, and this entire process will be referred to as the enzyme inhibition. Okay, now this enzyme inhibition is in both ways. It is beneficial for the organism if it is regulatory in function, or if it is extrinsic, like say if we are using some kind of drugs or antibiotics or some poisons are affecting us, so in that case it may also be harmful. Okay, now, most of the drugs, whatever we use to treat different kinds of ailments or different kinds of diseases in human beings, I'm not talking about only about infections. Non-infectious diseases also, specifically those which are associated with some sort of biochemical pathway. So in that case, these inhibitors may be utilized to treat that particular condition or at least to control that particular condition. आप इन inhibitors को आसानी से use कर सकते हैं. Now, the inhibition process itself. So we have basically two kinds of inhibition processes or two kinds of inhibitors. One we refer as the reversible inhibitors. Another category is irreversible inhibitor. As the name suggests itself, reversible means one which can be reversed, means activity can be retained, and irreversible means whose activity cannot be retained. Now again, in reversible inhibitors, they have been classified as competitive, non-competitive, and uncompetitive inhibitors. Now, competitive inhibitors, what are these competitive inhibitors? If we see the reaction here, this is your enzyme molecule, this is your substrate molecule. The reaction starts, it forms ES complex, it will be converted to product and the enzyme molecule. Now, when inhibitor molecule is involved, what happens? The reaction occurs in this way. Enzyme couples with the inhibitor molecule. A enzyme inhibitor complex is formed and substrate is left as it is. It cannot couple or it cannot bind with the substrate molecule. अब आपकी substrate आपके enzyme के साथ couple नहीं हो पाती. As a result, there will be no formation of this ES complex, and if this doesn't form, reaction cannot proceed. Okay, so here you could see that what is happening, that your inhibitor molecule is mimicking the substrate molecule with respect to the complementarity. If you see this substrate molecule, ये आपका substrate molecule है जो green green सा part दिख रहा है, and this pinkish part is your inhibitor molecule. So what you observe that to some extent this inhibitor is similar to this substrate molecule. Now as a result, what happens? It goes and attaches to the enzyme molecule, but since it does not have the exact resemblance with the substrate, somehow the reaction is blocked. चूंकि एक्चुअल में सबस्ट्रेट नहीं है तो जाहिर सी बात है प्रोडक्ट तैयार नहीं होगी. यू जस्ट कंसिडर दैट यू हैव अ लॉक, उसकी स्पेसिफिक की अगर वहां इंसर्ट करोगे तो वो लॉक ओपन हो जाएगा. नाउ सपोज यू हैव अ की जो थोड़ी सी ओरिजिनल की के जैसी नजर आती है, सो देयर आर चांसेस कि जब आप वो की इंसर्ट करोगे सो दैट लॉक मे गेट ब्लॉक्ड. वो चाबी वहीं पर फंस जाएगी, आपका लॉक बेकार हो जाएगा, आप फिर उस लॉक को इस्तेमाल नहीं कर पाते. सेम थिंग हैपेंस विद दैट इनहिबिटर मॉलिक्यूल. आप बिल्कुल एक टोटली डिफरेंट की अगर डालेंगे तो वो डालने के बाद कुछ भी नहीं होगा और वो की बाहर निकल जाएगी. लेकिन अगर वो दूसरी की ओरिजिनल की से थोड़ी सी भी रिजेंबल करती है, थोड़ी सी उसके जैसी कुछ चीजें उसके अंदर है, सो व्हाट इज गोइंग टू हैपन, आपने की डाली, थोड़ा सा घुमाया तो वो घूम भी जाती है और फिर कहीं जाकर के दरमियान में ब्लॉक हो जाती है. एज अ रिजल्ट अब आपकी ओरिजिनल की आप वहां पर नहीं डाल सकते क्योंकि जहाँ की डालना है वो जगह डुप्लीकेट की ने, दूसरी की ने ब्लॉक कर ली है. सो द सेम थिंग इज डन बाय द इनहिबिटर मॉलिक्यूल. हमारा जो इनहिबिटर मॉलिक्यूल होता है वो भी ठीक यही चीज करता है कि वो इस एक्टिव साइट को ब्लॉक कर देता है. एज अ रिजल्ट व्हाट हैपेंस, जब तक के ये एंजाइम सॉरी इनहिबिटर मॉलिक्यूल यहाँ पर है, सबस्ट्रेट मॉलिक्यूल वहां आकर के अटैच नहीं हो सकता. एज अ रिजल्ट द एंटायर रिएक्शन इज ब्लॉक्ड. पूरी की पूरी रिएक्शन यहाँ पर ब्लॉक हो जाती है. नॉन कॉम्पिटिटिव इनहिबिटर्स में आप देखेंगे सेम थिंग हैपेंस, देयर विल बी फॉर्मेशन enzyme and substrate will be there will be formation of this enzyme substrate complex product and enzyme will be freed up. But non-competitive inhibitions में क्या हो रहा है, as is evident from this figure that non-competitive inhibitor does not bind at the active site. ये एक्टिव साइट पर बाइंड नहीं होता है. सो इफ यू सी दिस प्रीवियस स्लाइड कॉम्पिटिटिव इनहिबिटर्स, यहाँ पर इनहिबिटर और सबस्ट्रेट मॉलिक्यूल इन दोनों के बीच में कंपटीशन होगा दैट दे हैव टू बाइंड एट द सेम पोजीशन one at a time. If substrate is attached, inhibitor cannot do anything, it cannot stop the reaction. Okay, अगर इनहिबिटर पहले अटैच हुआ वो सबस्ट्रेट को रोकेगा और रिएक्शन ब्लॉक हो जाएगी. बट इफ substrate goes and attaches to the enzyme molecule, then inhibitor cannot stop that reaction. This is competitive inhibition because both the substrate and the enzyme inhibitor molecule, they are fighting to couple at the same place, same location. Whereas in second case, non-competitive inhibition, the substrate does not couples at the active site. Instead, it has got another region on the enzyme molecule where it attaches. Okay. Now, even if there will be formation of this enzyme substrate complex, so here what you see that this reaction can be this enzyme inhibitor reaction can be carried out in two ways. First, the enzyme will couple with the inhibitor molecule, the EI complex will be formed, and the enzyme will become non-usable. In other case, when enzyme substrate complex is formed, it has to be converted to the product. But meanwhile, inhibitor molecule comes and that inhibitor molecule attaches at a particular location other than the active site of the enzyme, and as a result what happens, the conformation of the enzymes change. As a result, product is not formed, the enzyme substrate complex becomes inert. The enzyme further cannot convert the substrate to the product molecule. The reaction is blocked. Okay. So since now, if you want to compare this competitive and non-competitive inhibitors, how you can compare? Suppose this is your enzyme molecule, this is your substrate molecule. So this substrate comes, attaches with the enzyme. enzyme substrate complex is formed and some kind of product is formed and released. Okay, this is usual enzyme substrate reaction. Okay, when there is competitive inhibitor, suppose this is your substrate molecule and say this is your inhibitor molecule. So what is going to happen? There are two chances that either the substrate will go and attach with the enzyme, the reaction will occur. If this thing has happened, inhibitor will remain silent, it cannot revert this reaction, वो इस reaction को बदल नहीं सकता है. अब product form होगी, enzyme release होगा, तब कहीं जाकर के आपका ये inhibitor आकर के यहाँ पर attach हो सके. Okay, now suppose this is enzyme and this is inhibitor molecule, it comes and attaches here. So there will be no scope, no chance that the substrate can attach with the enzyme molecule. As a result, the enzyme will be blocked. In non-competitive inhibition, what is happening? Say this is your enzyme molecule. Now inhibitor comes and attaches at this location. As a result, the conformations of enzyme change. Initially, enzyme was like this. Active side of enzyme was like this. The substrate has, the inhibitor has came. It has attached. As a result, enzyme की structure बदल गई. अब अगर substrate यहाँ पर आए तो वो attach नहीं हो सकता. One probability. दूसरी कि ये सबस्ट्रेट आया यहाँ पर अटैच भी हो गया. लेट अस कंसीडर कि ये इस तरह से आपका एंजाइम मॉलिक्यूल है. अब यहाँ आकर के एक इनहिबिटर अटैच होता है, फर्दर इट चेंजेस द कन्फॉर्मेशन. सो एज अ रिजल्ट द एंजाइम सबस्ट्रेट कॉम्प्लेक्स विल बी लाइक दिस. द एंजाइम इज unable to convert the substrate molecule to the product molecule. Enzyme अब भी आपकी substrate को product में convert नहीं कर सकता. Okay, so when enzyme is coupling at the active site, sorry, the inhibitor is coupling at the active site, the reversible inhibition will be competitive. When the inhibitor is not coupling at the active side of the enzyme, it is coupling at elsewhere. Enzyme molecule के बाकी हिस्से में कहीं पर attach होता है, so it will be referred to as the uncompetitive inhibition. Non-competitive inhibition. And lastly, the uncompetitive inhibition. If you see now what this uncompetitive inhibition is, अब competitive inhibition क्या है? That the inhibitor binds only to the enzyme substrate complex. It does not have the capacity to bind to the free enzyme, and it cannot be overcome by increasing the substrate concentration. Now we'll see what these concepts are. एक अनकॉम्पिटिटिव इनहिबिटर जो है वो कब कपल होगा एंजाइम के साथ? जब एंजाइम ने सबस्ट्रेट के साथ एक कॉम्प्लेक्स तैयार कर लिया है. What we have seen in case of what we have seen in case of non-competitive inhibitors, that it can couple with enzyme in both the situations. Even if enzyme is free, even if there is formation of ES complex, in both conditions, non-competitive inhibition can attach to the enzyme molecule. But in case of uncompetitive inhibitors, there is condition that there should be formation of ES complex. As is evident from this particular figure, you can see this is the active side of the enzyme molecule, this, and this is the location where your inhibitor molecule is going to attach. यहाँ पर आपका inhibitor molecule आकर के attach होने वाला है. Okay. Now, what you observe here, that as soon as the substrate comes and attaches itself to this active site, there is a bit of change in the conformation of this site where inhibitor is going to attach. जैसे ही substrate यहाँ पर attach होगा, आप देखें यहाँ पर ये location कुछ इस तरह से थी, अब ये थोड़ी सी conformations इसकी change हो जाती है. Now, as a result, this enzyme is now available for attachment with the inhibitor molecule. The inhibitor molecule will come and it will attach with this enzyme substrate complex, and thereby it will block the entire reaction. Okay, so these are the reversible forms of inhibition. Competitive, where the attachment of inhibitor and substrate molecule is only at the active side of the enzyme. Non-competitive, where the attachment of the inhibitor and substrate are at different locations, but inhibitor can couple with the ES complex as well as the free enzyme. Okay. Uncompetitive means that inhibition where only enzyme substrate complex can react with the inhibitor molecule. Free enzymes will not interact with the enzyme molecule. Now here we have a comparison of competitive, non-competitive and uncompetitive inhibition. These graphs you will understand later when you study the enzyme kinetics in detail. Now as you could see here that the effect of competitive, non-competitive and uncompetitive inhibition on the enzyme catalyzed reaction, the rate of reaction is decreasing. यहाँ पर आप देखेंगे इन तीनों ही केस में when there is case of enzyme inhibition, the rate of reaction, the velocity, Vmax refers to the velocity of the reaction, decreases significantly. In competitive inhibition, this reduction is bit low, whereas in non-competitive and uncompetitive inhibition, there is significant decrease in the rate of reaction. Now, what is the role played by this thing in the cell's regulatory mechanisms? At the start, we have said कि cell इन inhibitory mechanisms को regulation के लिए use करता है, control switch के तौर पर use करता है, right? Now, these irreversible inhibitions, they act as the regulatory mechanisms for a particular cell. All these mechanisms, especially competitive and non-competitive inhibition, will be dependent on the substrate concentration and the product concentration. दो aspect पर ये depend करेंगे कि substrate का और enzyme का concentration कितना है. In all the cases, if the inhibitor concentration is higher than the substrate concentration, then the enzyme will be inhibited. If the concentration of inhibitor is negligible as compared to substrate, the inhibition will not be to that extent, because in competitive and non-competitive inhibition, the substrate concentration plays a major role. अगर आप की reaction में inhibition हो रहा है, competitive या non-competitive inhibition होता है, and if you simply increase the concentration of substrate, the inhibitor will become ineffective. The reaction will be carried out at the regular pace until the concentration of substrate is reduced from the concentration of inhibitor. Okay, and there is another way, the irreversible inhibitions. So those irreversible inhibitions, they permanently block that enzyme catalyzed reaction. The enzyme molecule becomes useless in entirety. Whereas in reversible inhibition, there are chances that by some method you can counteract the effect of inhibitor molecules and continue the reaction to be carried out. Irreversible inhibitions में आप reaction को further continue करवा सकते हैं by certain means, by increasing the concentration of substrate molecules in the reaction or by altering the conditions. There are certain methods for doing that thing which cell usually adopts. But in case of irreversible inhibition, the loss of enzyme activity will be permanent. The enzyme which has been coupled with the irreversible inhibitors, they cannot be reutilized. The cell has to synthesize fresh enzyme molecules if it wants to carry out the reaction further. Okay, so this much for the day. Irreversible inhibition and remaining things regarding the enzymes we'll see in your next lecture. Meanwhile, if anybody of you have any queries, we have about 3 minutes time, so you may ask your questions. आप आपके क्वेश्चंस, आपका अगर माइक अवेलेबल है, सो यू कैन आस्क थ्रू दैट, और इफ अनफॉर्चुनेटली यू डू नॉट हैव माइक, सो आप चैट बॉक्स में लिख करके आपकी क्वेरी रेस कर सकते हैं. यू कैन टाइप योर क्वेश्चंस. आई विल सी एंड ट्राई टू आंसर दैट.